In this video, we will discuss about the non-self and self Glycan epitopes. In our previous video, we have discussed how the therapeutic approaches can be used to using this non-self and self Glycan epitopes to target the tumor cells. And here we will discuss what are the what are the some of the what are here we will discuss some of the examples of the non-self epitopes and also this self-glycan epitopes, and what are the receptors or immune response that are already available to differentiate this non-self and self-glycan epitopes, and what are the artificially generated receptors can be used to differentiate this non-self and self-glycan epitopes. So first we will discuss about this self-glycan epitopes. So these are the self-glycan epitopes that are found on the mammalian cells that we can see here. This is the uh, we can say end-linked glycan carbohydrate structures that are present, and if there are non-self epitopes that are found here, such as these high mannose glycan epitopes, then this can be detected by this immune response such as these hyper-reactive T cells, and it will generate the pro-inflammatory response to kill this kind of cells on which this high mannose glycan epitopes are found. Similarly, this is also a high mannose glycan epitopes which are not usually found in the mammalian cells, and this can be detected by these anti-glycan antibodies, and these are the artificially generated and artificially induced antibodies, and these antibodies can be linked with this immune response, so that these kind of cells which are on which these high mannose glycan structures are found can be targeted. And simultaneously, apart from these T cells and these anti-glycan antibodies, this galectin 3 can also detect this glycan epitopes and can generate an immune response against the non-self glycan epitopes. That one of the examples that we can see here. And and one more receptors that we have also already covered in our previous video, that is C-type lectin receptors, which are usually found on the dendritic cells that is seen here. And these C-type lectin detectors can also detect the non-self glycan epitopes and can generate an immune response against it. As we can see here, this is one of the non-self glycan epitopes, that is very high mannose glycan structures which are found in the microorganisms, and it can generate an immune response by with the help of this C-type lectin receptors that helps in detection of these non-self glycan epitopes and generate an anti-inflammatory response. If this kind of glycan epitopes are found in the organism for a longer duration of time, that it might even leads to the autoimmune response, and that is because of that might trigger this anti-inflammatory response, which is usually arises there is a continuous uh, whenever there is a continuous generation of the cytokines that might cause damage to the tissues. And other types of other types of, and this kind of non-self glycan epitopes, that is very high long nanostructures, is usually found in the bacteria microorganism, and in fungi microorganism also has these high nanostructures, but it is distributed into the more number of branches that we can see here. Here the number of branches are low, whereas when the carbohydrates are generated in this fungi microorganism, the number of branches of high mannose glycan epitopes are more, that can be seen here. Such as this kind of glycan epitopes are found in this S cerevisiae, and it will generate the pro-inflammatory response whenever the dendritic cells recognize this kind of non-self glycan epitopes. And in the case of virus, these glycan epitopes are usually found on the spike structural protein, on the spike structural protein of the virus. That, for example, in SARS-CoV-2, it is found on the uh, it is found on the spike protein. It is found on the other structural part of these viruses also, but usually there is a spike. The glycans that are present on the spike region is is more prone to detection by this immune system of the host. In this case, the example shown is there. The DG sign can recognize the SARS-CoV-2 glycan structures, and in the case of HIV, these glycan structures are detected by this. Glycan structures on the spike proteins are detected by these dendritic cells, and it will generate the pro-inflammatory response. So this is some of the examples that are shown here, where in the case of viruses. And we can see in the case of viruses, the non-self glycan epitopes are completely different. They are not like in the case of microorganism with the high nanostructures. It's a completely different glycan structures, but they are not similar to the glycan epitopes that are found on the mammalian cells, but they may evolve themselves, and with time and with time, but as they replicate, SARS-CoV-2 virus replicates, they might generate the epitope similar to the mammalian cells. So it is essential to detect this kind of non-self epitope at the point of infections, because at that time the glycan epitopes, the replications of the SARS-CoV-2 might not, or SARS-CoV-2 or other virus might not be high, and they might not get enough opportunity to generate the glycan epitopes that are similar to the host organism. So this is how this non-self and self glycan epitopes can be differentiated by the host immune response. And as we go from this final figure is shown here is represents that if we go from virus to this we can say simple living organisms to complex living organism, we can say the glycan epitopes completely changes. And this glycans epitopes that are found in the simple microorganisms might not be suitable for this complex microorganism, and the immune system of the complex microorganism might not consider them as might consider them as harmful for the host cells, for the host organism, and try to kill those microorganisms on which these different non-self glycan epitopes are found. So that's how this immune response can be targeted, or immune response can be used to target this non to target this cancerous cells, and helps the immune response to differentiate between the cancerous cells and the healthy cells, and can increase and this strategy can also enhances the immune efficiency of the hosts. So I hope you have understood about some brief overview about this non-self and self glycan epitopes. If you need to understand this non-self and self glycan epitopes in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.